Right. So thank you for the introduction and thank you for hosting me. So as Shambhavi mentioned, I recently graduated from the Department of Electrical and Computer Engineering at Johns Hopkins with a PhD, and I currently work at IBM Research Lab at Almaden. My PhD thesis advisor was Dr. Venkatraman, and my doctoral work has centered around the broad theme of developing novel and powerful representational frameworks. And these frameworks have been primarily developed for application to computational neuroimaging data. And what we're trying to do here is leverage ideas from machine learning, optimization, and geometric and generative modeling. So from the clinical standpoint, we sought to bridge the gap between connectomics with that of behavioral phenotypes. And what we are looking at is a predictive setting. And our primary motivation was to improve our understanding of complex neurodevelopmental disorders, so particularly autism spectrum disorder, beyond just diagnostic classification. So we have been working closely with researchers at the Kennedy Krieger Institute at Johns Hopkins. And with that very small introduction, let's dive into the talk. So we live in a world that's naturally organized into intrinsic network structures. For example, at the biological scale of organization, we have the human genome. Here, various encoding proteins may interact with each other in complex ways, and these define characteristic human traits. On the other hand, the age of the internet has given rise to complex social networks such as the ones we see on Facebook. And recently, these have shown to have tremendous power in influencing people's decisions at an individual level. A wireless network is another example where various devices communicate with each other point to point. So the study of networks is very relevant to the modern world as we get a lot of insight into otherwise mysterious phenomenon. In fact, one such complex network is the human brain. And recently, there has been a lot of interest in understanding how regions in the brain communicate with each other and how these communication patterns influence our behavior and health. And this sets us up for a really important but challenging question. So the human brain is a vital organ that coordinates key information and plays a key role in decision making. Anatomically, it is a network of individual processing centers connected by neuronal axons. Functionally, we can think of the brain as partitioned into a myriad of regional hubs that specialize at complex and sometimes abstract tasks. In this work, we have explored this duality in representation via structural and functional neuroimaging data. So these are measured respectively by diffusion tensor imaging and resting state functional MRI acquisitions. And this figure illustrates the connectomics or connectivity-based views of function and structure, which we use in order to map brain organization. From the algorithmic and computational perspective, we have been witnessing an explosion in terms of data-driven methodologies. And this has largely been fueled by a phenomenal increase in compute power, along with curation of large data sets such as MNIST or ImageNet. In this regard, however, data sets for medical imaging lie on the complete other end of the spectrum. And this is particularly pronounced for neuroimaging studies, such as the applications that we discuss in this work. While a typical computer vision or speech dataset may have over tens of thousands of examples, what we are looking at here is of the order of 50 to 100 patients, which is a much reduced sample size. At the same time, this data has a lot of complexity in terms of the underlying interactions within the brain. For example, differences in brain connectivity could be very subtle, especially in disorders such as schizophrenia or autism. At the same time, aggregating data from multiple sites could also be challenging due to differences in acquisition and population demographics, as is common with many medical imaging studies. As a result, clinical studies remain modest in terms of dataset size. To summarize, there is often a lot of heterogeneity within patients, which many models fail to capture. So it is imperative to develop frameworks that can effectively tease apart individual differences from this data. And this is where my work comes in. So overall, I've organized my talk into 4 subparts. In the first 2 sections, I will build up frameworks that can jointly model functional and structural connectomics data and predict behavioral measures in clinical populations. The last 2 segments of the talk focus on geometric modeling extensions that leverage deep learning techniques for better understanding clinical and neuroimaging information. In the first section, I will describe a generative discriminative framework that we developed. This framework is designed to automatically extract brain networks from functional connectivity data that are predictive of behavioral measures. So in a typical resting state functional MRI scan, a patient lies still in the scanner while being imaged. The resulting acquisition is such that each volume element or voxel in the brain has a time series response that's associated with it. The underlying rationale is that brain regions which communicate with each other display synchrony in terms of their activation. To characterize these functional relationships, we often use correlation matrices. So these capture similarity between time series responses across different brain regions. For example, a commonly used measure is the Pearson's correlation coefficient that you see here, which makes these matrices symmetric along the diagonal. What we wish to infer from here is the latent functional organization in the brain. Further, what we want to do is to utilize this information to examine symptom severity that's associated with, say, a particular disorder. Often this symptom severity is a numerical measures that is scored by clinicians and typically encodes the level of impairment that a patient has in a particular facet. Now, a typical machine learning setup for this problem would say use the features corresponding to the upper triangular part of the symmetric matrix for each patient. So let's look at how a typical machine learning pipeline for this kind of analysis looks like. It's important to note that the vectorized correlation matrices that I described earlier are typically of the order of thousands, depending on what parcellation we choose for the brain. And this is fairly large compared to the number of patients in the study. Thus, a natural first step here would be to perform dimensionality reduction to compactly represent this information and retain salient information. Commonly, ICA or graph-based centrality measures or statistical approaches such as PCA may be used here. So once the representation is brought to a more manageable space, the projected features can then be fed into a discriminative model that predicts clinical diagnosis. Again, we could use a simple linear or a more complex nonlinear model. So while such frameworks have been widely used for this task, a major disadvantage is that these approaches are pipelined. So effectively what that means is that if our dimensionality reduction step is off, we cannot hope to get a meaningful representation for our downstream task. Additionally, most of the literature focuses on classification of patients versus controls. This effectively groups all of the patients into one class and performs some sort of a group level discrimination. But these disorders are known to be pretty heterogeneous, which means that although there are group level commonalities among all subjects, there are also subtle differences in how two different individuals manifest symptoms, and most existing frameworks fail to model these effects directly. So these limitations motivates the joint model that we propose, and our model overall has two parts. The first is a generative model, which simultaneously accounts for group level as well as patient-specific information from patient correlation matrices. The group level canonical representation can be mapped back into the brain and is shared among patients, as we can see from this cartoon here. From here, the patient-specific information that's extracted is fed into a discriminative regression framework to predict clinical severity. And instead of pipelining the two, what we are trying to do is to jointly infer the generative and the discriminative parts of the representation within one single optimization. So now that we have a high-level picture, let's first look at the blue box representation in more detail. So we begin with the correlation matrix for each patient, which is given by gamma n. The group level term here is a basis matrix B. The columns capture coactivation patterns in the brain that are common to all the patients. And this can be seen from the exemplar subnetwork here, as you can see in the columns of this matrix. Although the subnetworks are the same for each patient, the way in which they combine to explain the patient-specific correlation matrix are individual-specific. So we can capture this by a patient-specific diagonal network strength matrix, as is shown here. This combines the subnetworks using an outer product to explain each patient's correlation matrix. This dictionary decomposition is similar to common principal components and ties in with our motivation of capturing group level and patient-specific information simultaneously. Intuitively, what we are trying to do is to model the functional communication within the brain via distributed and spatially overlapping subsystems. Next, we will look at the discriminative model in the gray box. So using each patient's diagonal network strength term, we stack these coefficients into a matrix C. It has k networks and n patients. So each row of this matrix represents one patient. And typically we use a small number of networks that capture the information most faithfully, effectively reducing the dimensionality of the fMRI data that we started out with. From here, we then use a linear regression with a weight matrix W to predict clinical severity. This is a clinical measure per patient and is captured in the vector Y as given here. So these are the constituents of the joint model, and this work first appeared in MICI 2018, and an extended version of this is available in our 2020 NeuroImage paper. We refer to this as our Joint Network Optimization Framework, or JNO for short. In the following slide, we will mathematically formalize this. So we have a joint objective function with two main terms. The first of this is the matrix factorization, and the second is for clinical severity prediction, which was the gray box we saw earlier. We add an additional L1 penalty to the basis to encourage sparsity and highlight important brain regions. We add an additional non-negativity constraint on the Cs, since our correlation matrices are by construction positive semi-definite. We add additional L2 penalties to ensure that this estimation problem is well posed. So the inputs to the training model are the correlation matrices and the severity scores. The unknowns that we are trying to estimate are the coefficients, the basis, and the regression weights. Note that we are jointly trying to optimize these latent variables here. To simplify the estimation, we can notice that the subproblems in each of these variables can be tackled more easily. Hence, we develop an alternating minimization scheme with three main steps. For example, fixing C and W, we can use proximal gradient descent to update the basis, and this handles the L1 penalty. The objective is quadratic in C and convex, so we can utilize quadratic programming solvers. Finally, W has a closed form solution. So what we do is cycle through these steps until a notion of overall convergence is reached. To summarize what we are trying to do here, we are trying to obtain a joint representation that explains the connectivity data and clinical severity simultaneously. To evaluate this model, we have resting state fMRI scans collected from 58 patients with autism spectrum disorder, or ASD for short. As mentioned earlier, ASD results in diverse clinical impairments. In this study, we have validated our model on three separate clinical measures. The first of these is ADAS, which captures social and communicative deficits along with restricted and repetitive behaviors and is scored by a clinical exam. The SRS is another score that quantifies social responsiveness and is typically a parent or a teacher questionnaire. And finally, Praxis is a motor measure in which trained clinicians score individuals based on imitation, gesture to command, and tool usage. Overall, these measures quantify a wide range of diverse impairments that characterize the disorder and employ different methods of reporting. This again contributes to the heterogeneity in the cohort, which is actually what our models are trying to characterize. So we perform a k-fold cross-validation for our methods and the baselines, where we train the model on a percentage split of the data and estimate the model parameters, which in our case are the hidden variables in the model, and test on the unseen patients. Here we compare against two separate machine learning baselines. The first performs linear dimensionality reduction, followed by random forest regression. The second is a nonlinear dimensionality reduction followed by random forest regression. Both baselines emulate the pipelined approaches that were mentioned in the introductory slides. So we first look at the prediction of ADAS. So to the left is our model. PCA plus random forest is in the center, and kernel PCA and random forest is at the extreme right. In each of these plots, the x-axis describes the score given by the clinician, while the score as predicted by each model is on the y-axis. Thus, ideal performance should be given by the x equal to y line. The red points indicate training fit, and the green ones denote prediction on unheld out testing data. For the training data, all of the models fit pretty well. However, when it comes to unseen data, the baselines fail to generalize. In fact, they are not able to detect differences between patients and typically concentrate around the mean. Unlike this, our method is better at capturing trends in unseen patients, and this is also quantified by the differences in median absolute error and mutual information. The performance difference can also be seen on the SRS prediction in terms of both qualitative and quantitative performance. At the same time, our method isn't perfect either, as there are some outlying points. Again, this should be expected in a sense, given that the score reporting itself can contribute to some of this variability. We observe a similar performance for praxis. Overall, all of these performance profiles indicate the difficulty of the prediction task itself. While all methods have the representational capacity to fit the training data, we conjecture that the baselines lack the flexibility to learn representations that are directly informative of the measure that we are trying to predict. In contrast, our optimization procedure by design is designed to uncover a representational space that is directly informative of behavior, because all of the representations are jointly optimized. We have more comprehensive comparisons in our neuroimaging paper, where we have compared this framework against several graph theoretic machine learning and statistical feature selection and regression models. Apart from the predictive performance, we can also examine the coactivation patterns present in the basis B. So we illustrate 8 subnetworks learned here on our model, which is trained to predict ADAS. With the help of our clinical collaborators, we were able to identify the learned patterns and correspond them to networks seen commonly in the ASD literature, including the default mode network or somatary sensory and visual networks. So the main takeaway here is that the learned representation is actually of clinical relevance. It offers interpretability as opposed to being black box. Further, we can compare and contrast the patterns learned for predicting various measures. For example, we illustrate one of the 8 subnetworks for predicting ADAS to the left, SRS to the middle, and praxis to the right. This comprises of regions from the default mode network, which has widely been inferred in the autism literature. While we see that there is a high level of overlap in the network, though they are trained across different scores, on one hand, it indicates robustness of the procedure, since the framework is trained on essentially the same scans in each case. On the other hand, the subtle differences in the basis patterns help adapt to the prediction of individual measures. Taking a step back, this overlap suggests a common canonical representation, which can be useful for mapping to a complete spectrum of behavioral deficits. Along these lines, a natural extension that we tried is to train the model on multi-output linear regression. However, this did not perform as well on predicting multiple deficits at the same time. This is a technical limitation that motivates part 2 of my talk. So to summarize what we have seen so far, our model has introduced a common generative basis which captures group level information and also acts as a dimensionality reduction. Our discriminative coefficients capture heterogeneity in the population, while our joint optimization helps extract generalizable and interpretable representations. Now, some of the limitations we would have liked this model to address are listed here. First, we would like to obtain flexible representations which can explain multiple behavioral deficits simultaneously. And this would help in a more holistic characterization of such complex disorders. We would also like our model to exploit complementary anatomical connectivity information. We can think of this approach as using prior domain knowledge in the form of structural connectivity to guide the functional representation. Lastly, our framework examines connectivity in the brain as a static snapshot that's averaged over the entire scan. But literature suggests that functional connectivity is a dynamic process that evolves over time and space. And modeling this evolution can help improve our understanding of the functional organization in the brain. So with that brief motivation, let's move to the second part of the talk. Here, we extend the JNO framework by utilizing techniques from graph theory and deep learning to build up a deep generative hybrid model. So first, we take a step back and can take a second look at functional connectivity. So over the course of the scan, it is believed that the brain toggles between different cognitively identifiable states. We represent this by this GIF here. We see that the quote-unquote states cycle through phases of activity and relative inactivity. This switching profile is unique to each individual and is believed to be clinically informative. This is why we are interested in modeling this evolution. Prior work in this area builds upon techniques such as HMMs that can encode and unravel transition between such latent states. Other approaches include dynamic conditional correlation or the graphical lasso, which propose various mathematical formulations to estimate this connectivity. Most of these frameworks model interactions between large subsystems that are grouped a priori in the brain. And these may not scale very well to whole brain ROI level analysis. We circumvent this problem by adopting a generative model from the JNO to study sliding window correlations and estimate the underlying latent profiles. Structural connectivity, on the other hand, is estimated using diffusion tensor imaging, or DTI. In a nutshell, it utilizes the directional diffusion of water molecules in the brain to estimate fiber tracts that connect individual regions. Using cryptography, we can thus construct detailed anatomical maps of the brain. Thus, structural connectivity would provide information that's complementary to functional signaling. There has been an increasing interest in integrating these two representations, particularly for diagnostic applications. This is how a typical data-driven multimodal integration pipeline would look like. Often, such approaches would fall into two broad themes. Some of the techniques separately analyze the statistics of individual modalities or extract graph theoretic features separately, and then draw correspondences between the features post hoc. Such approaches may not directly exploit the complementarity between the two representations. Another common set of models combines the modalities in the same feature extraction module using data-driven techniques. Once these features are obtained, a discriminative model can then be trained on them. Such approaches may suffer from the same limitations as discussed with the pipeline machine learning models in part 1. We would like to address these limitations directly at the modeling level. Therefore, we propose a joint multimodal generative model. This integrates the fMRI and DTI connectivity together at the feature representation level. We also couple the generative model with a deep regression framework into a single optimization. Pictorially, we can illustrate these ideas here. The blue box describes our multimodal representation learning scheme, while the gray box describes the deep network that maps to behavior. So let's look at the generative model in more detail. So we begin with the dynamic functional connectivity matrices, so gamma and P. Again, we have a basis B, which is common to all patients and is shared across time as well. Each column captures shared coactivation patterns, which can be mapped back to the brain. The way in which these subnetworks combine, now over time, is unique to each patient. We capture this using a temporal network strength term, C and T. We combine these subnetworks in an outer product fashion to estimate the dynamic functional correlation matrices. Our generative model is called the SRDDL, or Structurally Regularized Dynamic Dictionary Learning. So it builds onto this base framework. The DTI tractography matrices describe graphs that are given by anatomical connectivity between regions of the brain. In fact, it's believed that the functional relationships are mediated by either direct or indirect pathways. Therefore, we would like to use this to guide our decomposition. We model the interplay via a structural graph regularization based on the graph Laplacian operator. And mathematically, the Laplacian is calculated by a scale difference between the degree matrix and the adjacency matrix that we obtain from tractography. The predictive part of our model is a deep network that's given by the gray box here and maps to behavior. We first vectorize the diagonal contributions from the K network coefficients, as is shown in the figure. The K networks are the rows and the time points are the columns. Per subject, these column vectors C1 through CTN feed into an LSTM network, and we want to track the temporal evolution and estimate an embedding that's informative of behavior. Thus, this embedding H is input to a two-branched ANN consisting of a predictor ANN and an attention ANN. The predictor ANN uses the intermediate representation to provide time-varying estimates of clinical severity vector Y. So Y now captures multiple impairments in a single patient. Since the dimensionality of our inputs is fairly small, we use a simple neural network with two hidden layers. At the same time, we would like to automatically pick out salient regions in the scan that are important for prediction. Hence, we use temporal attention. Essentially, the attention ANN learns a scalar weight A over time from the embeddings to combine the prediction estimates. Overall, this deep network is deliberately designed to help track which time points over the scan are especially relevant for clinical score prediction. These are the components that form our deep SRDDL model, and this work was first presented in NICAI last year. An extended version appeared this year in NeuroImage. Let's mathematically formalize this setup. The objective has two main terms. The first is the SRDDL loss. Mathematically, our structural regularization replaces the classical L2 norm by a graph Laplacian regularizer to provide anatomical weighting. Here, we made use of the weighted Frobenius norm with the weighting as described by the graph Laplacian. While the standard isotropic loss would equally weight all functional connections, what we are trying to do here is to reweight our estimation according to the structural graph. Next, our deep network loss is the standard MSE loss between the outputs of the ANN and the target severity scores, and is a part of our joint objective. We add an additional non-negativity constraint on the coefficients since the gamma NTs are positive semi-definite. The orthogonality constraint on the basis promotes uncorrelated patterns of activation in the columns of B that explain the connectivity data well. To recap, the inputs to our model are the correlation matrices, the DTI graph Laplacians, and the clinical severity scores. What we want to jointly estimate are the basis, the dictionary coefficients, and the deep network weights. This is a more difficult inference problem than before, but we can nevertheless simplify the inference by looking at the sum problems in alternating minimization. This has the three main steps as listed here. B has a closed form Procrustes projection solution. Unlike a conventional optimization of the coefficients, we can instead resort to a stochastic optimization using gradients estimated by backpropagating all the way to the inputs of the network and combine these with the parametric dictionary learning gradients. The weight updates are standard for deep models, and we cycle through these three steps until a notion of overall convergence is reached. So we can note here that the updates for the coefficients is the key step. It effectively balances the representation of the neuroimaging data with the generalization to prediction of behavior. We again evaluate on the same ASD cohort, now with DTI and fMRI scans. Our clinical scores are the same as before, except that the model is now trying to jointly predict the complete severity profile for each patient instead of just the individual measures one at a time. We use a cross-validation strategy and evaluate against two classes of baselines. So the first baseline is an adaptation of the BrainNet CNN that was introduced by Kawahara et al. It can operate directly on the connectomes without vectorizing by using specialized cross-shaped convolutions. This baseline has two branches, one for the DTI and one for the fMRI static connectomes. The representations are combined to predict behavior. Our second baseline is more data-driven. We use PCA to reduce the dimensionality of the dynamic correlation features and impose the structural weighting by a Hadamard product. The projected data is used to train an LSTM-ENN model similar to our framework and predict clinical severity. The main takeaway from these comparisons is that the first baseline incorporates the network structure and multimodal interactions, but ignores the temporal evolution. The second baseline incorporates multimodality and temporal dynamics, but not the network structure of the inputs. So here's the performance profile for the BrainNet CNN baseline. Note that we are now trying to jointly predict the three measures. We have ADOS to the left, SRS to the middle, and Praxis to the right. In each subplot, the score as given by the clinician is on the x-axis, while the score as predicted by the model is on the y-axis, with the diagonal line indicating ideal performance. Red denotes training points, while blue indicates testing points. Similarly for the PCA plus LSTM baseline. First, we see that the baselines do fit the training data faithfully and are able to generalize to the ADOS score prediction. In fact, the PCA baseline does really well on ADOS. However, this is at the expense of predicting the other two, because at test time, the models hover at around the mean and sometimes even do worse than mean prediction on the other scores. Finally, only our method is able to obtain a viable trade-off between prediction of all the scores and provides improved performance in terms of median absolute error and mutual information. Empirically, we observe this trend with this as well as other baselines in our comparison, where they can be tuned to predict one of the three measures really well, but this is always at the expense of generalizing to the remaining measures. Our model, though not perfect, is successful at estimating a representation that can faithfully map to different attributes within the behavioral space. Overall, this performance profile suggests that this is an extremely challenging prediction paradigm. Another advantage of our framework is the interpretability in the representation. As an illustration, here are 4 of the 15 subnetworks, and we note that they have been widely inferred in the ASD literature in the past, and are known to be relevant to the clinical impairments our model is mapping to. And this is a key advantage compared to conventional deep learning, because we can actually probe the representations. Moreover, we can also examine the temporal dynamics that the model encodes. For example, here, we look at the attention weights in our deep network. The attention weights for each of the patients at testing time are plotted as a heat map, and our model is designed to implicitly handle scans of different durations, as is seen by grouping the patients with shorter scans in the beginning. First, we note that the learned patterns concentrate on specific areas of the scan and tend to be sparse. They highlight the start of the scan for some patients while focus on the end of the scan or middle for others. This alludes to the insights on the underlying patient heterogeneity. Additionally, we can look at the network strength C of a representative patient as it evolves over time. In this plot, each solid line corresponds to one of the 15 different subnetworks. Here, we see that some networks become active and relatively inactive over the scan. This closely mirrors the transient brain states hypothesis in the resting state literature. In fact, the switching profile is unique to the individual and can be compared and tracked across patients, which may be useful for making comparisons between subtypes of the population. Overall, the main contributions of the model are: we were able to incorporate structural information in the form of anatomical priors to guide our functional connectivity. Our deep network allowed us to track temporal changes in the scan, and the optimization procedure helped generalize to predicting multiple impairments. This blend of utilizing domain knowledge combined with the flexibility of deep networks helps us obtain a more holistic picture of brain connectivity and behavior. At the same time, we would like to address some of the limitations of this formulation. For example, the graph-based regularization may not paint the full picture of the interplay between function and structure. We are interested in capturing complex topologies within brain connectivity better. An example of this topology could be the hierarchy of organization. And at the same time, we would also like to work on improving predictive performance. We explore some of these ideas via end-to-end geometric deep learning models that are the central focus for the rest of the talk. In this section of the talk, we deviate slightly past our original goal of predicting phenotypes. Instead, what we are interested in further exploring is the complementarity between functional and structural connectivity data spaces. So resting state fMRI and DTI provide us with dual representations of the brain, and each of these is endowed with its own set of geometric properties. These are two complementary aspects of brain connectivity, which are distinct but intimately related. In a bid to better understand this relationship, we pose a slightly different question from the phenotypic prediction. Instead, what we are interested in predicting here is predicting one view of connectivity from the other, and we focus on the prediction of structure from function. At the same time, understanding the role of clinical information and its relationship to connectivity provides a window into challenging translational paradigms. Therefore, we would like to have our representation be clinically informative. So keeping this in mind, we use behavioral information as a secondary guide in our representation learning. Here is the pictorial outline of the matrix autoencoder model that we developed. Our model consists of three main components, one for representation learning in the functional connectivity space. Effectively, this performs reconstruction of input functional connectivity matrices. The second component maps from function to structure, that is, it predicts structural connectivity from functional connectivity. The third block maps to phenotypic measures. In the next slide, I will introduce the representational framework for functional connectivity. So as seen previously, the functional connectivity matrices lie on a low-dimensional manifold of symmetric positive semi-definite matrices. And we design an autoencoder representation with self-supervision to reflect these properties. This leverages the known parallels of autoencoders with classical decompositions in the low-dimensional setting. Our encoder takes as input the functional correlation matrices, and we make use of 2D FCNN layers that were introduced in Dong et al. These encode a canonical outer product decomposition that can be parameterized by the weight W. From here, the decoder creates a reconstruction of the gamma n's using the same weight as the encoder. Overall, this autoencoder estimates a symmetric low-rank matrix decomposition from the input correlation matrices. The weights learned in the columns of W can be thought of as canonical brain bases for functional connectivity. Unlike the frameworks we built up in previous sections, the autoencoder allows us to solve for a common principal components like decomposition, but this time within a neural network optimization architecture. So let's examine the function to structure prediction block in more detail. So we use a second decoder to map the low-dimensional functional connectivity embedding F to the structural connectivity matrices A using a cascade of 2D FCNN layers. We would like to use an alignment decoder consisting of 2D FCNN layers to map from the functional to structural embedding. So our structural connectivity matrices capture the number of fiber tracts connecting different pairs of brain regions. And by construction, these matrices are hollow and standardized to have an Elman norm 1. In order to represent this within the model, we suppress the diagonal entries of the decoder output and apply a softmax at the end to the off-diagonal elements. Finally, we have a third branch for prediction of phenotypes from functional connectivity. We use the projections X as inputs to an ANN that predicts the vector Y. The component first performs a 1D convolution on the projected FC features And this allows us to do a forward pass through a cascade of fully connected layers with leaky relu activation. Overall, this prediction branch serves as a secondary regularizer for the matrix autoencoder model and encourages the representations to be informative of clinical characterizations. This slide illustrates the complete framework that appeared last year in MICAI. To train the joint model, we adopt a stochastic Adam optimizer and use a separate validation set for tuning. For this study, we evaluate on two separate data sets with fMRI and DTI scans. The first is a set of healthy controls from the HCP database, while the second is our in-house data set of patients with autism. For the purposes of evaluation, we use functional connectivity matrices obtained from average regional time series. And for the DTI data, we use outputs of tectography, that is, the estimates of the fiber track between regions in the brain. On the behavioral side, the evaluation on the HCP is for predicting a single score that measures cognition, which is the Cognitive Fluid Intelligence score, while for the autism dataset, we use the same phenotypic measures as before. In each case, we adopt a five-fold cross-validation strategy. First, we evaluate the prediction of structure from function. In this plot, we visualize the FC and SC embeddings using a t-SNE plot. We observe well-separated point clusters between the two modalities, and this separation hints that the model is successful at differentiating the two views. What the alignment decoder is learning is a mapping between these two sets of features. Next, we can evaluate the structural connectivity prediction against two key baselines. For each model, what we are comparing is the distance of the test examples with the ground truth structural connectivity against the average distance from other examples. For a good reconstruction, we expect F self, that is, the distance between the distance from the ground truth, should be an average lower than F other, which is the distance from other examples. We see this reflected within our framework. The first baseline is a regularized linear regression between the FC and SC features, which we see performs worse than our model. The second uses a matrix autoencoder with the structural connectivity as input and at the output. This is a proxy for the best case performance. In this sense, our model performs only slightly worse than this scenario. Overall, these results indicate that the matrix autoencoder learns a stable transformation between the two data spaces. We can also see very similar trends with our evaluation on the autism dataset. Next, we examine the phenotypic prediction task. Our first set of comparisons are against three ablated versions of the model, and these help us evaluate individual components, such as the utility of the resting state fMRI decoder. Second, the effect of removing the DTI prediction. Third, the effect of combining the connectivity representation with behavioral prediction end-to-end in a neural network framework. We also compare against two deep learning baselines that are designed for the prediction task. The first is the multimodal version of the BrainNet CNN, while the second is the deep SRDDL from part 2 of this work. Looking at the results on the HCP database, we observe that on the test examples, our framework outperforms all of the baselines in terms of three quantitative metrics. This suggests that the captured biomarkers are also predictive of clinical measures. At the same time, the performance gains against ablated versions of the model indicate the utility of the autoencoder formulation, the function to structure mapping, and the joint representation learning. We also see that the model generalizes well to the prediction on the autism dataset and provides comparable performance against state-of-the-art baselines. This is, in fact, a more challenging paradigm than single score prediction, as we are trying to predict multiple deficits simultaneously. We can also examine the basis representation that is learned by the matrix autoencoder. And to examine the robustness, we can match the learned basis across different cross-validation folds and initializations, and display the most common set of patterns on an average. We observe that the regional standard deviations here tend to be small for a majority of the region, indicating that the extraction of functional biomarkers is fairly robust. Moreover, we can also identify several commonly observed patterns that are relevant to cognition from the HCP database and which have been previously identified in studies. Thus, the learned representation is of clinical consequence as well. We can make similar observations with the KKI dataset as well. And thus, our model allows us to probe the learned representation and is not completely black box as opposed to existing deep learning methods. To summarize our contributions from this section, we have developed a matrix autoencoder that can exploit the underlying geometry of the functional connectivity. Our structural connectivity alignment decoder learns a robust mapping from function to structure, and the phenotypic prediction serves as a secondary regularizer. Overall, this model generalizes well onto unseen examples, both for phenotypic prediction as well as function to structure prediction. This framework elegantly combines the interpretability that's offered by classical decomposition models, along with the simplicity and flexibility offered by deep learning. In relation to the other 2 points we alluded to earlier, the final part of my talk will shift focus away from models inspired by classical decompositions. In the final section, we deviate from classical representations based on exploiting low-rank matrix structure. Instead, we focus on the network-centric view of brain connectivity by utilizing a different kind of geometric model, which is the graph neural network. A natural way to look at brain connectivity is network-centric. This explicitly focuses on the graph structure between communicating regions. Here, each node of the brain graph could correspond to regions within the brain, and relationship between pair of regions are the edges that connect these. We could additionally have node-based or edge-based attributes as inputs. From the data-driven perspective, graph neural networks, particularly graph convolutional networks, have become a popular tool for analysis. A typical GCN layer consists of multiple filters that can aggregate connectivity information within the nodes. Composed together, the layers can underscore pathways of information propagation within the graph, as can be guided by a downstream task such as case control prediction. While there are several works which utilize graph convolutional networks to predict diagnosis from brain connectivity, for example, from the fMRI data or from DTI, these models typically use the graph structure encoded by spatial or connectivity relationships. Often the connectivity profile is vectorized within the graph signal, rather than utilizing the network structure within the data itself. These models typically consider one modality at a time and focus on group-wise comparisons. Instead, what we'd like to do is to fuse information from functional and structural connectomes using carefully designed graph filtering operations and map to fine-grained prediction rather than classification. This is an outline of the multimodal graph convolutional model, or the MGCN for short. The model consists of 2 main parts. The first is the representation learning, which utilizes graph convolutional feature extraction, which is the green box. The inputs are the functional connectivity matrices and the DTI structural connectivity matrices, while the second part is a regression model that maps to phenotypic measures. Let us look at the graph-based representation learning first. From the fMRI data, we have connectivity information as represented by a correlation matrix of size P cross P. Complementary to this, we have cryptography-based structural connectivity matrices from DTI, which inform us about anatomical connectivity within the same ROIs. Our model assumes that the latent brain graph structure for a patient is given by the DTI graph Laplacian L, and the correlation matrices gamma n are the associated graph signal. We utilize a topological graph filtering operation at each node pair within the graph. So mathematically, if WR and WC are the row and column filters, we can represent the filtering operation by this equation here. At a high level, this computes a weighted sum of the functional connectivity as regularized by the DTI Laplacian. This uses a first-order approximation to graph filtering, similar to Kipf and Benning's 2017 paper. The filtering operation also mirrors the E2E filters in Kawahara et al. And simultaneously considers both the row-wise and column-wise connectivity information. Next, we use a second graph convolutional operation with pooling at the end to obtain a node-wise representation H2, which can be mathematically summarized as follows. So this is similar in flavor to the graph centrality measures in graph theory and summarizes node-wise information within the graph. Last, we can perform a third graph convolution embedding using a fully connected layer with weights G. These are what we input into the regression model. A regression model is a simple 3-layered ANN with fully connects and leaky ReLUs. The outputs Y can be either a single or a multi-output measure of phenotypic information. This is our overall architecture, which we train using a combination of L1 and L2 losses. Empirically, this provides a good trade-off between stability and generalization between different regimes in the loss landscape. Our model parameters were set using a separate validation holdout to infer the hyperparameters, and this work recently appeared last year in the Middle Conference. We evaluate on 2 fMRI and DTI scan sets, i.e. the HCP and our in-house KKI data set for the autism patients. As mentioned before, our evaluation on HCP is a single score prediction, while on KKI, we evaluate on multi-score prediction. We again utilize a five-fold cross-validation and report on the performance against a couple of baselines. So the first two baselines can be thought of as ablated versions of the MGCN. The first is a multimodal ANN with the correlation features and the structural Laplacian features as inputs. The multimodal information here is fused via a hard-mod product, and we designed the ANN to have the same number of learnable parameters as our MGCN, and this helps us evaluate the benefit provided by using a GCN as a feature extractor. The second baseline uses the same architecture as the MGCN, but removes the contributions of the DTI graph Laplacians. This helps us evaluate the benefit of adding structural regularization to the representation learning. The third baseline is the multimodal BrainNet CNN, which has two branches. And our final comparison is the deep SRDDL model, which we introduced previously. Overall, these last two baselines evaluate the benefit of using GCNs for implicit representation learning over other deep learning alternatives. This is the performance on the HCP dataset. We can observe that for the test examples, our framework outperforms all of the baselines in terms of qualitative as well as quantitative measures and suggests improved generalization. On the KKI dataset, we examine the ability to jointly predict all three measures, that is ADOS, SRS, and taxes. We observe that our framework clearly outperforms the ANN, the RS-fMRI only GCN, and the BrainNet CNN baseline on the multi-score prediction task on all three phenotypes. Our model also outperforms the deep SRDDL, though the performance differences are a little less dramatic. This also illustrates the benefit of utilizing the network structure of the connectomes for prediction. As such, generalizing the prediction of multiple measures is inherently really challenging. This is indicated by the performance trends comparing single versus multi-output regression in the previous and current slide. Overall, this framework is an important first step to developing end-to-end models which generalize reliably on small data sets. To summarize this section, our multimodal GCN helps integrate functional and structural connectivity data. Our filtering operations leverage topological information within the brain graph and help us extract representations that are clinically predictive. This framework shows promise at generalizing on two data sets for predicting behavioral information. So since we are at the end of the talk, here's a brief highlight of the main contributions. Based on the insights from this work, we have identified a couple of promising directions, which from the algorithmic standpoint, we can examine the problem of multi-site representation learning using larger data sets and aggregating such such multi-site data can be challenging due to distribution shifts or site-specific biases, and which our current models may not be robust to. Similarly, from the clinical standpoints, our frameworks could be used to study the prediction of fine-grained behavioral scores from connectivity data. But these studies could not provide us with the complete clinical picture. For example, the prevalence of different subtypes within the disorder or comorbidities, and data-driven analysis in this domain could be challenging. So this is another line of important direction for exploration, where we can develop frameworks that can be designed to uncover more nuanced characterizations. Before concluding, we would like to acknowledge these funding agencies, and I'm indebted to these several researchers without whom a lot of this work wouldn't have been as meaningful. This presentation described work that appeared in these sets of publications. And thanks for listening to me, and I'd be happy to take questions.
Great.
I'm sorry.
Yeah. Is there an echo from my side? I think all of this covered all your thesis, right?
Mostly, yes.
Yeah. Is your thesis available online?
Not yet, but it should be available around me.
Okay, great. I have questions beyond the talk as well. And if anyone has to ask any questions, please feel free to put in the chat box, and we'll let you unmute yourself and you can ask. But what I'm curious about is how did you get to the topic of your research during the PhD? Was it pre-assigned, or did you have to find a way through it?
So is this part of the talk also recorded?
I can stop the recording. That's okay.
Sure. If it's general questions related to PhD research, then maybe you can stop the recording because it could go in a